Is by Mohamed Fahmy from Rostock University on functional analysis of miRNA transcription factor synergistic regulatory motifs in Parkinson's disease.
Hello, everyone. I would like to thank the organizers for giving me this nice chance to present my recent research work. So as a motivation, I'd like to start with this slide. So one of the many challenges of modern systems biology and precision medicine is data integration. So right now, everyone tries to describe the problem from his expertise. So the one who works on gene expression data comes with some candidates for biomarkers. Similarly, also people who are working on the genetic variations also identify some genetic variations. But in fact, all of these are just putting their hands onto one or two pieces of puzzles. And in order to gather or to unravel the whole picture, so one has to cover all these pieces of puzzles together in order to identify the whole picture behind the pathogenesis of diseases. So inspired by that, I'd like to present to you what I have implemented, so an integrative network-based approach, and I have implemented it and demonstrated its applicability and efficacy on Parkinson's disease paper, on Parkinson's disease. So at the beginning, so I start with collecting microRNA samples and gene expression samples from Parkinson's disease patients as well as normal patients, and then starting with the data pre-processing and doing the differential analysis. So ending up with deregulated genes and deregulated microRNAs, and now trying to integrate all these deregulated microRNAs and genes into a complete transcription factor microRNA regulatory network. And from this network, then I'll try to extract the functional subnetworks which are related to the pathogenesis of Parkinson's disease, and also identifying or detecting specific recurrent network motifs between a microRNA and a gene as well. And doing as an end some validation using statistical analysis and also semantic validation, as we will see later. And beside that, after identifying these functional modules, then I'll try to connect them to the links and CMAP drug databases to identify some inhibiting molecules which can be used in order to normalize the apparent expression of the, or induced by the disease itself. So the, the framework itself was encapsulated into our web server, which is called TFMER. So this TFMER web server can be used in order to construct disease-specific GRN network from microRNAs and transcription factors and co-regulated genes together. And as, as you see here, as you see here, it starts with the deregulated sets of microRNAs and genes, and then pooling all the different interactions, transcription factors regulating genes, microRNAs regulating genes, transcription factors regulating macroRNAs, and if available, microRNAs also regulating microRNAs. There are some evidences behind this fact, and then collecting all of that into one big network, which is called TF, microRNA network, and then extracting this or subtracting this network into a disease-specific network and doing some standard network analysis afterwards. And I'd like to show you here some results of the data on a Parkinson's disease paper. So on the side here, on the left side, you see the heat map for the deregulated genes. And as you see, they were able to cluster the disease samples and the normal samples almost perfectly. And also here, the Parkinson's disease associated genes and microRNAs were found to be totally deregulated here with a significant level. As you see, we used also TFMER in order to construct the Parkinson's disease gene regulatory network, which is composed of microRNAs highlighted here in yellow, and also some other nodes, transcription factors, and other co-regulatory genes. And we applied some centrality measures in order to identify the corresponding driver networks here, nodes. And then from these nodes, or from this network, we identified the functional modules as we explained at the beginning. So for example, the neurogenesis module, the regulation of apoptosis and dopamine metabolism, and also identified the recurrent network motifs here and checked afterward the functional homogeneities between these networks or subnetworks using the similarity measure between their functional association. So, for example, here we show the similarity or the significance of the gene similarity or functional similarity between the gene pairs of this motif and also the gene pairs of randomly selected genes within the network. Also, we use these functional modules in order to identify the possible drugs trying to normalize the changes or the abnormal changes of the abnormal changes in the expression induced by the disease itself. Then, aside from Parkinson's disease, we can also couple this approach into or extend it into a larger approach or into a larger radiogenomic approach. So we have applied this also into a prostate cancer, into cancer genomics, integrating this with clinical data as you see here, and also imaging data in order to identify or try to predict the pathological stage of prostate cancer patients. So from here, we used our approach in order to identify these sub-networks. And from the sub-networks, we have identified potential biomarkers, 3 microRNAs and 1 gene. And then we use these biomarkers beside with the clinical data in order to predict or better predict the pathological stages of prostate cancer patients. And interestingly, we found also that the expression profiles of these biomarkers were found to be highly correlated with the radiographic features extracted from multiparametric MRI images here. This is the GRN of prostate cancer, and this is my last slide, by the way. And then the GRN of the prostate cancer, and also the biomarker expression levels. And finally, here is the prediction using 2 different machine learning algorithms. And as you see here, the combination of the clinical data and genomic biomarkers were found to perform better here with, compared with the clinical data alone. With this, I would like to conclude my talk, and I would like to thank the DAD for supporting my travel here, and also I would like to thank the funding organizations behind these projects, and thank you for your attention.
Okay, and with the quick talks, we don't try and squeeze in questions, so I would encourage you to talk to the speakers in the coffee break. Our next talk is by George Aquamensa on HER2, difference in transcriptional regulation between breast cancer molecular subtypes. And he is from the Massachusetts College of Pharmacy and Health Sciences.